Circulation. Circulatory pathway through the heart. Superior and inferior vena cava. Right atrium. Right ventricle. Pulmonary arteries. Lungs. Pulmonary veins. Left atrium. Left ventricle. Aorta. Body. 3 portal systems. Blood travels through an extracapillary bed before returning to the heart. Liver, hepatic, kidney, and brain, hypophyseal. Fetal circulation. Foramen ovale, connects right and left atria. Ductus arteriosus, connects pulmonary artery to aorta. Along with foramen ovale, shunts blood away from lungs. Ductus venosus, connects umbilical vein to inferior vena cava, connecting umbilical circulation to central circulation. Blood components. Plasma, aqueous mixture of nutrients, wastes, hormones, blood proteins, gases, and salts. Erythrocytes, red blood cells, carry oxygen. Hemoglobin. 4 subunits carry oxygen and carbon dioxide. Iron controls binding and releasing. Oxygen-hemoglobin dissociation. Factors leading to right shift of curve. Increase in temperature. The Bohr effect. Decrease in pH and increase in carbon dioxide. Oxygen release to tissues enhanced when hydrogen ions allosterically bind to hemoglobin. Increase in carbon dioxide leads to an increase in concentration of hydrogen ions. Leukocytes, white blood cells, function in immunity. Platelets, clotting. Platelets release thromboplastin, which, along with cofactors calcium and vitamin K, converts inactive prothrombin to active thrombin. Thrombin converts fibrinogen to fibrin, which surrounds blood cells to form the clot. Blood typing. Antigens are located on the surface of red blood cells. Blood type A. Red blood cell antigen A. Antibodies anti-B. Donates to A, AB. Receives from A or O. Blood type B. Red blood cell antigen B. Antibodies anti-A. Donates to B or AB. Receives from B or O. Blood type AB. Red blood cell antigen A and B. Antibodies none. Donates to AB. Receives from all. Blood type O. Red blood cell antigen none. Antibodies anti-A and B. Donates to all. Receives from O. Blood cells with Rh factor are Rh positive. These individuals produce no anti-Rh antibody. Rh negative blood cells lack the antigen. These individuals produce an antibody if exposed. Respiration. Gas exchange. Exchange occurs across the thin walls of alveoli. Deoxygenated blood enters the pulmonary capillaries that surround the alveoli. O2 from the inhaled air diffuses down its gradient into the capillaries, where it binds with hemoglobin and returns to the heart. Carbon dioxide from the tissues diffuses from the capillaries to the alveoli and is exhaled. Fetal respiration. Fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. Gas and nutrient exchanges occur across the placenta. Immune system. The body distinguishes between self and non-self. Humoral immunity, specific defense. Active immunity. Antibodies are produced during an immune response. Passive immunity. Antibodies produced by one organism are transferred to another organism. B lymphocytes. Memory cells. Remember antigen and speed up secondary response. Plasma cells make and release antibodies which induce antigen phagocytosis. Cell-mediated immunity, specific defense. T lymphocytes. Cytotoxic T cells destroy cells directly. Helper T cells activate B and T cells and macrophages by secreting lymphokines. Suppressor T cells regulate B and T cells to decrease anti-antigen activity. Memory cells remember antigen and speed up secondary response. Nonspecific immune response includes skin, passages lined with cilia, macrophages, inflammatory response, and interferons, proteins that help prevent the spread of a virus. Lymphatic system. Lymph vessels meet at the thoracic duct in the upper chest and neck, draining into the left subclavian vein of the cardiovascular system. Vessels carry lymph, excess interstitial fluid, and lacteals collect fats by absorbing chylomicrons in the small intestine. Lymph nodes are swellings along the vessels with phagocytic cells or macrophages. They remove foreign particles from lymph. Lipid digestion. When chyme is present, duodenum secretes the hormone cholecystokinin, CCK, into the blood. CCK stimulates the secretion of pancreatic enzymes and bile and promotes satiety. Bile is made in the liver and emulsifies fat in the small intestine. It is not an enzyme. Lipase is an enzyme made in the pancreas that hydrolyzes lipids in the small intestine. Enzyme. Salivary amylase, ptyalin. Production site. Salivary glands. Function site. Mouth. Hydrolysis reaction. Starch to maltose. Enzyme. Pancreatic amylase. Production site. Pancreas. Function site. Small intestine. Hydrolysis reaction. Starch to maltose. Enzyme. Maltase. Production site. Intestinal glands. Function site. Small intestine. Hydrolysis reaction. Maltose to 2-glucoses. Enzyme. Sucrase. Production site. Intestinal glands. Function site. Small intestine. Hydrolysis reaction. Sucrose to glucose and fructose. Enzyme. Lactase. Production site. Intestinal glands. Function site. Small intestine. Hydrolysis reaction. Lactose to glucose and galactose. Protein digestion. Enzyme. Pepsin. Production site. Gastric glands or chief cells. Function site. Stomach. Function. Hydrolyzes specific peptide bonds. Enzyme. Trypsin. Production site. Pancreas. Function site. Small intestine. Function. Hydrolyzes specific peptide bonds. Converts chymotrypsinogen to chymotrypsin. Enzyme. Chymotrypsin. Production site. Pancreas. Function site. Small intestine. Function. Hydrolyzes specific peptide bonds. Enzyme, carboxypeptidases A and B. Production site, pancreas. Function site, small intestine. Function, hydrolyzes terminal peptide bond at C-terminus. Enzyme, aminopeptidase. Function site, intestinal glands. Function site, hydrolyzes terminal peptide bond at N-terminus. Enzyme, dipeptidases. Production site, intestinal glands. Function site, small intestine. Function, hydrolyzes part of amino acids. Enzyme, enteropeptidase. Production site, intestinal glands. Function site, small intestine. Function, converts trypsinogen and procarboxypeptidases to active form.